Hello. Oh, there we go. Can we get seated, please? Thank you very much. So my name is Melissa Spearing with the Canadian Forest Service and the National Tree Seed Centre. I'll be moderating this session. So this is session 2A for genetic conservation. So I'd first like to introduce Yueru Zhao from a PhD candidate at UBC, who's going to present on predicting fundamental climate niches of forest trees based on species occurrence data. Okay.
Okay. So, sorry. Okay. Hello, my name is Yueru Zhao. I'm a PhD candidate from UBC from Dr. Tony Wang's lab. So today my work I'm presenting is predicting fundamental climate niche of forest trees based on species occurrence data. So I will go through the introduction and then the knowledge gaps and show the methods and results and then wrap everything up in a summary. So forests are essential in mitigating climate change, but they're facing new challenges. So we can see that climate niche is the range of climate conditions that trees are adapted to. And due to warming, the climate niche are shifting away. And then the trees, there will be a mismatch between the climate that the trees are historically adapted to and the trees will experience in the future. So climate niche models have been used to assess the impact of climate change of forest trees and then have been used for assisted migration. So if we know the patch below, if we see this is the current climate niche and the dashed patch is the future climate niche. And then we can see that for this part, we will need the conservation. And then for this part where we gain the new climate niche, assisted migration should be proposed. So in order to do conservation and assisted migration, we will need credible predictions of climate niche distribution. And climate niche models are built on forest species occurrence data and the historical climate conditions. And then with the future climate projections, we are able to predict forest species' future suitable climate conditions. So normally, these climate niche models predict binary classification maps. So let's say here are the original output from climate niche models. They should have cell values ranging from 0 to 1, representing the probability of the trees occur. But conventionally, traditionally, we would use a threshold at 0.5 to convert this probability map into this binary map. So for the cells having the value above 0.5, they will be considered presence for the trees. And then the cells having value below 0.5 will be considered not suitable, so absence for this tree. So there is a potential for us to use climate niche models to predict species fundamental niche by adjusting this cutoff threshold lower than 0.5, because if we're using the 0.5 threshold, the current prediction will be considered realized niche, which is the climate range that is limited or already restricted by biological and physical factors like competitions, landscape fragmentation, human disturbances, or dispersal limits. And essentially, realized niche is a nested subset of a species' fundamental niche. And it is, so species fundamental niche did not consider any of these factors. So it is the theoretical range, climate range that species can adapt it to. So for better climate change impact assessment and for a better range of assisted migration, it's better for us to know the distribution range of a species fundamental niche. So here is an example. So let's say if the blue curve is the realized climate niche, and then the red curve is the species fundamental climate niche. So we can see that in assessing the climate change impact, if the trees are left outside of their realized niche, but they're still within their fundamental niche, then they can still grow. But if we do not know the fundamental niche range, we would consider the trees here all die. And from another view, let's say if this patch is the realized niche distribution, but actually the fundamental niche range is like much larger both end. So if we're doing a system migration from here to here to suit into the future climate niche distribution, but without knowing the fundamental niche, we would consider that we're system migrating the trees beyond their native range. But actually, they might still be within their fundamental niche. So for a system migration, it would be less risky. And so here are the knowledge gaps. So the demand for the fundamental niche models and the fact that there was no fundamental niche model based on occurrence data. Because in doing so, we need the species occurrence data being introduced beyond their native range so that we can validate our prediction of the fundamental niche. And luckily, we have lodgepole pine and Douglas fir. So here are their distributions across the globe, not only because they're ecologically and economically important, but also they've been introduced into many other places. So they're native to Western North America, and then they've been introduced into Europe quite a lot, and even Southern Hemispheres. So with these two species data, we want to evaluate and compare various occurrence-based climate niche models and determine an optimal cutoff threshold to predict a species fundamental niche, and then apply this model to assess the climate, for the climate change impact assessment on the existing forest and explore the assisted migration potential. So the three models we used and compared in the study include Maxine, random forest, and generalized additive model. So here's the flow chart of how this study was done. We first did the presence to absence ratio optimization on the original native observation data. And then we optimized the climate variables. And we did the performance evaluation on all 4 candidate models, including 3 individual models and an ensemble model that combined the 2 top performed model from these 3. And then we use the global observations to select the final candidate model and adjust the cutoff threshold. So we will have a newly identified cutoff threshold, which can be used to predict species fundamental niche. And then with the original 0.5 cutoff threshold, we predict the species realized niche. So here is our results. Originally, normally, the presence to absence ratio, so which is the number of presence data points and the number of absence points you use in training your model. So normally, the ratio between these two are 1-to-1, because so that in this way, they are equally contributing in this model. But in our study, we tried to find a new presence to absence ratio for each algorithm. So we use the sensitivity and the specificity values to choose this ratio for each model. And we can see that for random forest, the optimal presence to absence ratio is at around 1 to 0.5 for both species. And for Maxine, it's at around 1 to 0.3 for lodgepole pine, and it's at 1 to 0.4 to Douglas fir. And then for generalized additive model, so there's no crossover point between the sensitivity and the specificity. So we can see that it's implying that generalized additive model generally overestimate the presence and underestimate the absence in the prediction. And Then we did evaluation to all the candidate models based on their AUC values and the other 3 metrics. So here we can see all the results showing that the ensemble model, which is the combination between the random forest and the maxine, had the overall best performance. And then with the global observations, we again calculated the ensemble model's sensitivity and specificity values and find the crossover point at around 1.3 of the cutoff threshold. So we decided to use this 0.5 cutoff threshold as our threshold to convert probability map into species fundamental niche map. And here comes these spatial prediction. So we can see that both species fundamental niche has much larger geographic extent. So the lighter blue color is the fundamental niche distribution, and the darker blue are the realized niche. So we can see it's much bigger for both species. And we also predicted the future niche loss on existing forests. That's why it's within Western North America. And so current distribution are the 3 colors together. And then if we do not know the fundamental niche loss, then the realized niche loss will be the color red and blue together. So that's quite a lot. But knowing the fundamental niche range, then we can see in future climate, the fundamental niche loss is actually much less. So it's actually a good news for us for conservation. And we also calculated the future niche gain. If the current realized niche is in the green color, and then the realized niche gain is in red color, which is very limited, but we can see the fundamental niche gain is much larger. So in summary, in our study, we found that the presence to absence ratio is critical in developing occurrence-based climate niche models. And then ensemble model of machine learning had the best performance. In our case, is the ensemble of random forest and maxine. And fundamental niche model would reflect more realistic niche loss or for climate change impact assessment and provides more potential for assisted migration. And then this fixed 0.3 cutoff threshold, which is consistent between lodgepole pine and Douglas fir, can be applied to other species if they do not have, they have not been introduced beyond their native range. So that's all from me. Please feel free to bring up any questions.
She made up for, oh, what's that? Oh, get away from the speaker, okay. Thank you for also helping make up for a little bit of time, but yeah, we've 2 minutes for questions, I think, and we'll.
Sure, I'll start. Why not use the exotic position, or the exotic planted forests in your training data? So you're basically, what I'm understanding is you're just allowing the native model to bleed a little further. But why not explicitly model it with the data from Europe and wherever the species are planted?
Sorry, so the question is why I only use native data for model training. So that's why that's why the cutoff threshold can be applied to other species if they are not introduced beyond their native range, because we're only using the data within their native range for the model training, and then the data beyond their native range are used for validation so that we know with the native range data we can also predict their fundamental niche.
Yeah, thank you for the presentation. And I wanted to ask this question to Tong Li yesterday, and you have spoken to the same problem, and he's your supervisor, so he can help you with the answer. Oh, no. So in your attempt to find the fundamental niche, there are two things that were brought in here. One, in the native range of the species itself, Douglas fir and the lodgepole pine, you have the component where the species is not necessarily its distribution is not necessarily limited by its inability to tolerate temperature or precipitation, but there is an ecological component where the distribution is limited by the species competition because it's occurring within the mosaic of other species around. So it's a competitive principle issues. And on one hand, you have also included the data to where the species has been introduced as exotics, like in Europe, and in Oceania, like in New Zealand, where the species actually is successful because it is grown in an environment where it is protected from competition. So how do these complicated two systems actually affect what you think is a fundamental niche? Okay.
I'm not sure if I get it correctly, but you mean that because the model is based on the natural forest data, but we're also using the plantations from Europe for validation?
Yes, I'm saying in the native range, so the species is not just the occurrence is not necessarily an indication of intolerance of the environment in there. It's just because the species may not be in there because there is more competitive species in that area. So then that describes your native environment. And in the exotic part, like in Germany or Sweden or New Zealand, the species is growing there as a single species where it is protected from competition because it is being farmed like any other garden in there. So when you put those systems together, how do they affect what you consider to be your fundamental niche?
For the data points within Western North America, they are natural forests. That's why it's reflecting a species realized niche because the competitions and all the physical and historical and biological factors are already included. But for the trees in Europe, they've been manually introduced beyond their native range. So that's why we believe that their occurrence means the distribution of the fundamental niche.
Okay. Sorry, Sean, we'll have to hold. Okay, so we're going to move on to the next speaker. Thank you very much, Yueru. I'll take your thing. There you go. So next we have Dr. Hayley Tumas, postdoc from UBC, to talk about genetic diversity in BC reforestation.
Yeah, so thank you. I am Hayley. I'm from UBC, which sits on the unceded and ancestral territory of the Musqueam. But I will be talking about research that takes place across the province, over 90% of which is on unceded and ancestral First Nations territory. And although I get the privilege of presenting on this work, it is a highly collaborative project, and we have collaborators from the Ministry of Forests, Mosaic, and Western Forest Products, in addition to the University of British Columbia. And we also have funding from Genome BC and the Forest Genetics Council. So here in BC, a lot of our breeding programs are beginning to increase in our breeding generations at the same time that a greater proportion of the landscape is being harvested and then replanted with the selectively improved material coming out of the breeding programs. And it's important to remember that these forests are a mosaic of natural and planted stands. And so this has begged the question as to what the impact of this forest management is on landscape level genetic diversity, especially because genetic diversity at this scale is going to be incredibly important for resiliency to climate change going into the future. And so here in BC, genetic diversity is already monitored and there is policy on genetic diversity. We have to meet a minimum effective population size of 10 in order for it to be actually planted out in the landscape. And so what is effective population size? It is the number of individuals in an idealized population experiencing the same rate of genetic drift, which is a really difficult concept even for population geneticists. And so the key here is the rate of genetic drift. And another way to think about it is that if we had a population of totally unrelated parents, that produce the exact same number of offspring, then your NE would be equal to that number of parents, which means that NE, or effective population size, is influenced by things like the number of parents, the relatedness of your parents, and the evenness of parental contributions. So all things that go into orchard design and breeding programs. And currently here in BC, NE is estimated for seedlots using parent tree gametic contributions, where you take pollen and cone counts for parents and get an overall estimate for any of that seedlot. But this policy and a lot of the work is still focused within planted stands, where we want to look across planted stands and at the landscape. And this is due to something that has been observed in fisheries, where it's common practice to take a small number of individuals from a natural population and use these as parents in captive breeding, where you produce a large number of offspring that are then used to demographically and genetically supplement your natural population. But it's been found that this actually decreases the overall effective population size. And this is down to the Ryman-Laker effect, where you have an increase in inbreeding and reduction in the total NE due to a small number of parents producing a large number of offspring, which sounds like something similar that might be happening here in forestry. So we want to know if a similar phenomenon is occurring in these selectively bred natural forests when we look at NE on a broader landscape scale. And so we're addressing this question over 3 activities that are going to look at different scales and stages of forest management. And so the first is looking at genetic diversity in seedlots, where we are comparing genetic diversity in this selectively bred material from the breeding programs to genetic diversity in seed collected from natural stands, as well as comparing genomic estimates of effective population size to those indirectly estimated currently for monitoring. And this first activity is using genomic data sets that were thankfully already available, in large part thanks to Ian McLaughlin, who collected this data as part of adaptory and co-adaptory projects for lodgepole pine and interior spruce. And I'll be showing you 3 different estimates of effective population size. So the first is that boots on the ground estimate, where we're using gamete production, using male and female contributions for each parent tree, and then estimating across the orchard. And then the other 2 are genomic estimates. So the first is linkage disequilibrium, or LD-NE, which is based on this idea that when alleles at unlinked loci become increasingly correlated, that means that your effective population size is decreasing and genetic drift is increasing. And the other is a lot easier to conceptualize. It's SIB-ship frequency, or SFNE. And so here we're doing pedigree reconstructions based on the idea that as relatedness among your samples increases, your NE is decreasing. And so we do have this nice large-scale data for lodgepole pine and interior spruce, but I'll just be showing you results for lodgepole pine because the results between the 2 species were quite similar. And so here we're comparing natural seed in green to that selectively improved orchard seedlot seed in blue for expected heterozygosity, so a measure of genetic diversity on top, and LD-NE, so a genomic estimate of NE on the bottom. And we have our estimates bounded by 95% confidence intervals. So you can see that expected heterozygosity is really not different at all. But we really begin to see these differences emerge when we look at effective population size, this much more sensitive metric, where we have our natural seed having much higher estimates with either the estimate or the upper confidence interval and infinity, which means that there was so little linkage we couldn't even get an estimate, represented here by those diamonds. And so this does mean that although genetic diversity might not be seemingly to decrease in heterozygosity in our breeding programs, genetic drift is occurring at a faster rate than in natural populations. And so on to that second question, where we're comparing our gamete production estimates in orange to our genomic estimates in green and blue. And we have our BC policy minimum NE of 10 here, so we see that all estimates are above that minimum. You can see that gamete production is quite similar to our genomic estimates, and that in most cases, it's actually lower. So this is actually really good news, at least from a genetic diversity standpoint. It means that anything that's getting planted on the landscape definitely has an NE well above 10. And it also means that the current way to estimate NE is actually doing quite a good job. And so thanks to our SIB-ship frequency estimates where we're doing pedigree reconstructions, we can actually kind of dig deeper to better understand what's going on here with effective population size. And so we can look at parent contributions. So the program would predict what parents exist based on the genotypes of the offspring. And so although we don't know exactly which parent this is in the orchard, we can look at parent contributions for each predicted parent. And so you can compare a higher effective population size seedlot to one with a lower NE. So you can begin to see that skew in parental contributions and understand that might be one of the things that's driving the lower effective population size. And the nice thing about this project is that we were able to add genomic data, and so we went out and genotyped most of the parents in the orchards that would have produced these seed lots. And so now we can go from having just a bunch of dummy parents to understanding which parents are actually making these contributions. So we have any parent that was actually genotyped and present in the seed orchard when these seed lots were produced in purple. And then the dummy parents, this is a parent that the software knows exists because each seed does actually have to have a mom and a dad, but it wasn't genotyped, is in green. So this could be pollen contamination, or it could be something else going on in the pipeline of both forest management and genomics. And so this also helps us to understand relationships across seed lots. And so any parent tree in red is shared between both orchard B and C. And the highest producing parent for our sample of the seed lot in orchard B is actually the mother of the second highest producing parent in orchard C. And so this means that as we move forward in forward selection, not only will there be greater relatedness within seed lots, but there will also be greater relatedness across seed lots, which is something important to think about when we're thinking about landscape level planning. It means that your planted stands on the landscape will also be increasingly related, which will affect broader scale effective population size. So then for the second activity, we're looking at genetic diversity in stands. So we're really looking at the shift here across stages from orchard to the seed lot that's produced to planted stands that are approaching crown closure in order to understand the impacts of things like natural regeneration, competition, and mortality on genetic diversity actually on the landscape. And so for this, we actually produced new genomic data sets for coastal Douglas fir. And so we sampled 2 orchards, each of which produced a seed lot, each of which was planted across 2 stands. And we sampled these stands at about 15 years of age. And so first, we're looking at the shift in genetic diversity from orchard parents in yellow to the seed lot it produced in blue to the stands in red. And you can see that, again, the expected heterozygosity on top was really showing no differences, but our LD&E is. And so our orchard parents have much higher effective population size, which is not surprising if you know what goes into this orchard design. A lot of these parents would have been plus trees from natural stands, and they would have been pulled from all over, so they do have high genetic diversity. But when we dig into the offspring, so the seed lots and the stands, we can see that our stands actually have higher genetic diversity than our seed lots. And how can that be? Because these stands are meant to be from the seed lot, so how do you gain genetic diversity on the landscape? This is most likely due to natural regeneration, where when you harvest an area and replant, you can have trees of the same species surrounding that stand, and so you can have seed influx and then seedlings, and they end up growing to be around the same size, so you really can't tell the difference when you're just looking at trees out in the landscape as to what came out of your breeding program versus what came from the landscape. But genomics can answer this question. So if we do a pedigree reconstruction, and we have an individual that has 2 parents, or at least 1 parent that is coming from the orchard, if you have those parent genotypes, then we know that that's a seed lot tree. And then if you have any seed that has neither of its parents are from your orchard, then that means that it would hopefully be a natural regeneration tree. Makes sense, at least the mom should be in the orchard. And so we can get some back of the envelope estimates of natural regeneration in these stands. We just take the number of offspring that don't have any orchard parents divided by the total number of trees that we sampled to get a rough estimate of the percentage of regen. And so now when we look here, we can see that Hillcrest, which has that highest effective population size, also has the highest amount of natural regeneration. And so we can then also exclude those trees that didn't have any orchard parents and recalculate effective population size. And now we see that our stand estimates are much closer to our seed lot estimates of NE, which does also then mean that natural regeneration is buffering genetic diversity in these planted stands, which is, again, good news when we're thinking about genetic diversity, but we're not entirely sure what impact this would have on genetic gain. I mean, a lot of work goes into producing these seed lot seeds, and so it'd be interesting to know this in the future. But we can, from this project, get a better understanding of natural regeneration within stands. So we collected 150 trees from each stand, and we did these along transects, which was difficult, at least for some of us who have never done forestry field work before. And we took a GPS point at each tree that we sampled, and so we can actually identify where in the stand natural regeneration is occurring. And so here we have one of the stands that was located on Vancouver Island, in these nice straight lines because John Degner from the Ministry of Forests and his team sampled this one. And we have seed that we know is a seed lot source because it has at least 1 orchard parent in yellow, and then any tree that doesn't have any orchard parents is most likely natural regeneration in orange. And so then we can also see how this varies geographically, because we did collect stands from multiple locations on Vancouver Island and then one on the mainland in Abbotsford. And so we can see how the natural regeneration might shift based on geography or based on site characteristics, because these did vary based on what was surrounding the stand and the stand itself. And so all of this is then leading up to the third activity, because you're like, okay, these are still looking within planted stands. How do I get to the landscape level? And so this is really what our third activity is building up to. We're looking at how might forest management be impacting landscape level genetic diversity. And to get at this broader scale, we're using a landscape genetic population simulation model, CD Metapop, which is a demogenetic model, which means that we can control both demographic parameters as well as genetic parameters. So we can vary parameters in order to perform a sensitivity analysis and really start to understand what factors might most be influencing genetic diversity. And so we'll be modeling both planted stands, which we will initiate using genetics that we already know about seed orchards, and as well as planted stands that would be connected via gene flow, so using all that genomic data that we had from the first two activities, and then running these simulations forward in time and varying these parameters to really understand how forest management might be impacting genetic diversity. And the nice thing about this model is that it is spatially explicit in landscape level. And so each of our stands in the model have an actual latitude, longitude that corresponds to an actual place out in the world. And so we will be modeling for, to kind of simulate a type of coastal Douglas fir situation, a coastal landscape on Vancouver Island, TFL 37, and are able to then account for things like protected old growth areas or parks, as well as use data, actual spatial data on planting in order to get a most realistic simulation as possible. And so these models are unfortunately currently in progress. I know this is probably one of the more exciting parts of the project, but be on the lookout into the future. And so, yeah, to wrap up, what we know so far is that the current genetic diversity monitoring is quite accurate here in BC, which is great, but that drift is occurring at a higher rate in our breeding populations compared to natural populations, and that forward selection will mean that there is increasing relatedness among our seed lots, as well as that natural regeneration then is buffering what is actually being planted on the landscape. And so with that, I do again want to thank our collaborators, Sally Aitken and Nick Ukranitz are two of the PIs in this project. We do have collaborators, Alvin as well, Dave, Marie, Trevor, working from Ministry of Forests, and Annette and Bevan have given us some great input from industry. And so with that, I'll take any comments or questions now, chat with me later, or feel free to reach out.
Thank you very much. Your presentations are always so beautiful. So time for 2 questions. Oda.
Thank you for your presentation. The genetic diversity or the lack thereof in your orchard, was that based on open pollinated seedlings that you guys then looked at its genetic makeup?
I'm sorry. I think there's a
Can you just repeat it closer to the mic?
It's my accent.
No.
Was the material from the orchard that you determined its genetic diversity, was it created through open pollination?
Oh yeah, good question. Yes.
Yeah, I thought so. Because you probably can maintain orchard diversity through a controlled pollination program. So you force the diversity. You can do that.
Yeah. So, so the seed lots and the material that would have been presented here is from seed lots that are actually operational. So yeah, it would be open pollination within those orchards. But you are right, you could do controlled pollination. I think I'm answering your question, hopefully.
Yeah,
Okay.
so there is a way to manage that.
I'm sorry?
There is a way then to manage that.
Yeah.
Okay, so I'll go to Barb.
Here you are.
Thanks for your talk. It's another orchard question. The Doug-fir orchard that you went into, was it a third generation orchard?
Good question. So
John says 2.
Too, because I'm just, that control of that inbreeding, I mean, it's how you make those selections determines whether you have relatedness, if you pick more than one individual per family to put into the orchard, things like that. That is controllable, right? And can get worse with higher generation orchards.
Right, yeah. So it is going to be getting worse, and yeah. So Douglas fir is obviously one of the older ones here, and so it is in greater advanced generations.
Okay, thank you very much. So next we have Wenhuan Xu. So, no, that's yours. So that's your forward button. Yeah. So he's going to be talking about an assessment of the impact of climate change on an endangered tree species. So, welcome.
Hi, hello. Wait, okay. Hello, everyone. Good morning. My name is Wenhuan, and my supervisor is Dr. Tongli Wang. Today, I'm glad to be here to share my study. The topic is about the assessment of the climate change on endangered conifer tree species. I will go over introduction, mathematics, results, and discussion. So just imagine if the temperature of this room go up to 40 Celsius degree and no one is allowed to move or taking any clothes, no air condition, no fan, it would be extremely uncomfortable, right? Unfortunately, some conifer tree species now are experiencing such discomfort. They are often really large, old, they are hardly to move, and they are usually grown in higher elevation of the mountain. So climate change has caused a mismatch between their geography distribution and the climate niche, which has led to severe consequences such as increased forest fire, pest dieback, and even the loss of biodiversity. So it's very important to assess such kind of mismatch. To assess the mismatch between the distribution and their climate niche, we often use climate niche model, which is also known as species distribution model. But for endangered conifer tree species conservation, a final model is usually required. So not only climate factor is important, but also soil factor. And interspecific competition should be considered to have a better conservation strategy. But the traditional climate niche model typically overlook these factors. My study aims to develop a new approach that can combine all these factors. I choose Taiwan red cypress as a case study to explore my new method. The reason I use that is not because red cypress is endangered, but also it has a great ecological importance. It has been called God tree and divine tree in Taiwan, and it is one of the World Heritage. I did a little bit literature research, and I found over 70 studies that reported its ecological and economic importance, but none has predicted its future suitable habitat. In order to assess the competition potential for red cypress, I introduced another species that is longinus oak. For red cypresses, they usually grow at higher elevation, around 1,800 to 2,500 meters. But for oak trees, they usually live at lower elevation, around 500 to 1,800 meters. However, due to climate change, those oak trees, they might move up and take up space of the red cypress forest. Here's the example. Like when the succession occurs, some broadleaf species will take up the space in the conifer forest and then compete for the resources. Here's an example. There is a oh, sorry. The red one. So here's a picture that shows.
There's a button. It's just right under here. It's just covered up, but that's the
Oh, okay, got it. Here's an fallen cypress tree, and we can see it has been surrounded by a lot of small oak trees, leaving no space for cypress to regenerate. So the objective of my study is to build a climate niche model for red cypress, and also build another niche model, which is called soil niche model, and then combine those to have a dual suitability prediction. Then I introduced long glance oak trees to compare the competition potential, to assess the competition potential for red cypress. Combine those information, I can provide a comprehensive information for red cypress conservation strategy. So how did I measure the dual suitability? The first step is I build two models, and I generate two maps, which is one is climate suitability, another is soil suitability. To create final map, I overlaid these two maps and generate a new map, and then I use min function to overlay these two maps, which is assign the smaller value between soil suitability and climate suitability to the new grade. In this method, I can ensure that a location cannot be considered suitable if it's only soil suitable or climate suitable, but not both. To measure competition potential, I also have two prediction. One is the oak trees prediction, one is the cypress distribution prediction. Then I overlaid these two maps to find the overlapping area, and I label the overlapping area in purple. Then I extracted those purple areas and calculated the presence probability of oak trees. Because higher probability the oak trees will appear, that means higher competition that the cypress may encounter in future. So this can be used to represent the competition levels. Here's the results. For dual suitability, this is the, This is the climate suitability. This is soil suitability, and this is the dual suitability. Then I predict the future 100 years. As we can see, with the time, with the climate change, the suitable habitat is decreasing. And if soil is considered, the decline is even greater. And here's the competition results. So currently, there is no much overlapping between these two species, around only 20%. But with climate change, by the end of the century, the overlapping area will go up to 80%. So a lot of overlapping. And here's the competition levels results. As we can see, in the southern part, the cypress will encounter severe consequences than the north part in the future. My study has both academic and application value. For application value, my study can be used to predict the areas of cypress forest in the future. And also, I can classify a location about the suitability for planting red cypress. This can be used for better conservation management. From an academic angle, I use the min function to combine soil and climate, and this function can be used in a wider context. such as typology plus climate. And our study also provides new insights to refine niche model. We also had a field trip, and I selected few potential sites that have mixed forest with both oak trees and cypress. Then I enter into the forest and randomly pick up 10 trees, recorded their coordinates, and generate a spatial map, use our model to calculate their present probability. All this step is for validation our model, and the results indicates that our model is accurate. Here are some pictures showing that the red cypress require big space and enough sunshine in order to regenerate. Okay, here's the conclusion. Our study developed a new approach that can assess the impact of climate change on endangered conifer tree species. The novelty of our study is that the way we combine the soil and climate, we use mean function. And also, our study highlights that the competition the cypress will encounter in future will increase with climate change, which highlight which provides implications for conservation and for endangered conifer tree species, and also for niche model development. So I would like to take this opportunity to express my appreciation, my supervisor, Dr. Tongli Wang, and with his full support, this study has been published in Science of the Total Environment. I will also thank for the genetic coordinating team to host this conference. And I received the financial support from Asia Pacific Network and China Scholarship Council for this study. I also would like to thank the data and pictures that are from Taiwan Forestry Research Institute and National Ilan University. I would like to thank the administration support from UBC Forestry faculty. So, and I think that's all for me. Thank you very much.
Thank you very much. I haven't had to use my cue cards once. This is fantastic. So we have lots of time for questions.
Hi, thanks for the great presentation. I'm just really curious. I don't know Taiwan very well, but that area that you identified that's going to be climatically suitable in the future, that has low probability of competition from the oak species, is any of that currently under protected land management status, like parks or anything?
So you mean this one? You mean the climate and suitability, this one?
Yeah, I'm just wondering if that area that you identified, which is such an amazing recommendation for management, right? Is there any area within that that's protected, or is that land at risk, or what's the status of that area?
Sorry, last question is what?
Yeah, is there a good chance of survival there? Yeah. Or like, are there parks, or is it protected land, or is it at risk?
Actually, what I predicted is about the natural distribution of them. So my data is from the natural occurrence. So basically, we don't consider the data from like park or protective areas, but red cypress is already protected in Taiwan. No one can kill them. No one can lodge them. And yeah, but even like this, even in this situation, the cypress forest will be decreasing because climate change. This is my study focus on, is about a natural force and natural selection under climate change. So that's what we focus on, about how the natural climate change will affect their suitable habitat in the future. Yeah.
Did you factor in sort of the natural migration rate of the oak into this? Like, I don't know geographically how big those spaces are, whether that's like 5 kilometers, 50 kilometers, like how fast can the oak actually transition into that space?
So your question is about the migration rate of the oak trees, right? So it is a good question. So that's also I'm interested in. Like migration rate is a key point that we can assess the impact of climate change. But unfortunately, it's hard to determine because the dispersal speed is hard to determine. But for oak trees, it's really easy for them to migrate because their seed are very light, and they are easy to migrate upward because they are small and they're easy to move. But for cypress, they are very large and they are very hard for them to move. That's why they will move forward and take up the space and compete resources for conifer tree species. But it's hard to determine how fast they can migrate up to. There are so many factors that should be considered in order to determine the specific speed, their migration rate.
Actually, I have a short follow-up one. Is there a natural animal disperser for that oak in Taiwan?
Yeah, sure. Animals is a factor that can move the seeds upward. Yeah. It's also a consideration. Yeah.
Okay. No, great study. So, thank you, Wenhuan.
Thank you.
Okay, so up next we have one of my colleagues, Martin Williams from the Atlantic Forestry Center, to discuss butternut conservation, moving forward in a recovery plan. So that's forward, yeah. That's forward, reverse, and I think that's the laser pointer.
Under the tape. Under the tape. Oh, this one. Perfect. Great. Well, hello, everybody. My name is Martin Williams. I'm a research scientist working for the Canadian Forest Service in Fredericton, New Brunswick. And today I'll be talking about butternut. So here's a short outline of my talk. I'll start off with a little bit of a background, provide maybe some of the work that we've done on our conservation efforts, and then maybe a little bit of discussion on what we think would be needed to help protect the species in the future. So butternut is one of 21 walnut species in the world. There's only 2 that are native to Canada. The black walnut, or Juglans nigra, that is mostly found in southern Ontario. And then this species holds a high value, especially in the US, as a timber species where the core of the species is found. And we have butternut, which is native to eastern Canada, as well as the US, more specifically in Ontario, Quebec, and New Brunswick. It is a early successional hardwood species that is a minor component of our forests, and doesn't live very long, only to about 100 years, and is also considered one of the most winter hardy of the juglans species. Another walnut species that is worth mentioning is Japanese walnut, or Juglans lentifolia. Japanese walnut was introduced in the late 1800s in both the US and Canada, and can hybridize with butternut. So among the tree species that I just mentioned, there's only one, fortunately, that is at risk, which is butternut. It is listed globally under the IUCN, and also in Canada. In the US, it is not listed federally. However, 23 states have it listed as either vulnerable, imperiled, or critically imperiled. In Canada specifically, There was a recovery strategy that was prepared in 2010 with the focus of really the conservation of germplasm as well as screening for resistance. So because of its endangered status and rarity, butternut is not an extremely extensively traded commodity, but it still holds a high ecological value as a rich food source for wildlife. It's also been extensively used and is a very important species for First Nations communities, where the tree was used in the past as a food source for medicine, for coloring dye, and also for the creation of exquisite art pieces, exquisite carvings like the one that you see here on the right that was produced by master carver Ned Bear. Ned Bear has passed, but this was a creation of his mask that was put on the cover of the Journal of Forestry. So Ned Bear as well was also a member of the Wolastoqey First Nation community in Fredericton, New Brunswick. So what is the issue with butternut? Why is it listed? Well, the problem we have with butternut is that they're infected by an exotic fungal pathogen called OCJ for short. The pathogen was first reported in 1967 in Wisconsin, in the US. But there have been reports that it could have been missed and could have been there prior, earlier than that in the southern US. The pathogen is responsible for butternut canker disease, which infects many tissues, leaves, wood tissue, even impacts seed. What we usually see in terms of symptoms is crown dieback, as you can see on this picture, and also cankers around the tree trunks and branches and limbs. And what we see is during the spring and in the summer, these cankers usually exude a black tar-like substance. This is really shows the presence of this canker. So what happens is that once infected, the disease usually ends up girdling the tree through multiple cankers, and then the trees die. So bigger trees takes a longer time to girdle, sometimes can take longer to die, but seedlings that are infected usually die off pretty quickly, and to the point that in our stands today, in natural populations, you don't see a whole lot of seedlings or recruitment into mature trees. So the pathogen has spread quite quickly throughout the range. It's now present all the way to New Brunswick, which is considered the most northeasternmost part of butternut's range and is disjunct. It was found there in 1997, and 20 years later, between 2014 and 2017, we resurveyed the population and found that the canker was also present across the range in both watersheds in the province, with a canker incidence rate of around 75%. Now, at that time, what we found is that the trees were still pretty healthy, with about 55% that had the higher vigor ratings of 4 in the province. So right now, what we're doing is we're going back, actually this year, we've started the surveying to see what was the impact after 6 to 7 years of that work. So what can we do from a conservation standpoint? Well, conservation usually happens in 2 ways, either by in situ conservation, which is protection within its own habitat. Unfortunately, the pathogen was not aware of our conservation policy, so it still infected the trees. So the other way is by ex situ conservation, and this is what we did for butternut. But there's only one problem with that as well. Butternut cannot store very well conventionally in the long term. So we proceeded with a cryo storage program that was led by Tanis Beardmore at the time. In short, the seed is collected and then dried to about 4 to 6% moisture content. And then once it's at 4 to 6% moisture content, we actually crack the nuts, extract the embryogenic axis, and put those in cryotubes and store in liquid nitrogen for long-term storage. So because we use that type of approach, we had to develop a different procedure to germinate, and we did that through in vitro germination to regenerate the plants back into trees. So last year, we recently tested a subsample of our collections that were done in the province. And what we're finding is that even after 7 years of cryopreservation, we still are maintaining the viability of our collection, which is great. In Juglans nigra, black walnut, they were able to keep it for 20 years. So we hope that that'll be the case for butternut as well. And just as an information piece, we have over 300 accessions collected from New Brunswick populations that are now stored in cryo as a backup. So having ex situ collections, I mean, hey, they're great, but it still does not slow species decline on the landscape. So what kind of options do we have for the future? Well, we have no registered cure. But even if we did have a cure, it's a good to have, but really limited to urban landscapes and to slow the decline. There's no biocontrol program to counter the canker. So really the only thing left is tree improvement. But as you all know, we need a source of tolerance to be able to do that. And the evidence of disease resistance in butternut is weak at best, if not absent. So what we do know is that butternut can hybridize with Japanese walnut, and both Japanese walnut and their hybrids are known to be more tolerant to the canker than the pure butternut. So it's likely that we will need some kind of Japanese walnut introgression to help protect the species, but there's a lot of work that needs to be done on that front. I mean, we're not as advanced as the American chestnut, and there are other factors that need to be taken into consideration to do this work, because as you know, all hybrids are not created equal. There's always issues that back crosses could dilute the tolerance, which was seen in the American chestnut, and there's other stressors. There are other biotic stressors that impact Japanese walnut more than they impact butternut. So we need to keep track and be aware of that kind of stuff. So what do we need? Well, like I said, there's a lot that's needed, but we're going to be focusing on doing some landscape genomics on the host and pathogen. But from a pathogen side, we're also interested in looking at strain pathogenicity to look at virulence and how it could impact the trees. And because we're looking at a possible hybrid strategy, it'll become very important for us to be able to characterize the individual trees to determine their hybrid status. So to do this, what we've done is developed a interspecific SNP panel with really the purpose of hybrid screening and with a focus really on separating our two congeners, butternut and Japanese walnut. This was developed using exome capture and GBS sequencing. And out of those sequences, we're able to find 34 nuclear SNPs and as well as 2 chloroplastic markers to design our panel. This is an example of 4 of our SNP selections, 2 that are specific to cinerea, so butternut, and in green here, 2 that are specific to Japanese walnut. We have a total of about 26 SNP markers to discriminate between these 2 species, and they're part of 15 of the 16 walnut chromosomes. And we have about from 1 to 5 SNPs per linkage group as part of the panel. And this work was all possible with the help of Manuel Lamotte, which is a biologist in Natalie's lab in Quebec City, as well as Aziz Abrimi, which is a postdoc in Douglas Jacobs' lab at Purdue University. So the first step in doing this interspecific panel was validating our SNPs. We basically selected 40 samples from pure butternuts that were collected from across the range to kind of look at whether the SNPs were fixed or not, what kind of variation we had. We also collected 16 samples of Japanese walnut, as well as various hybrids and backcross. And all of these selections for the validated panel were actually previously genotyped using either GBS, exome capture, or microsats that were produced in Sean Hoban's lab. So once we had that validation, we actually went out. There are orchards out there from putatively selected trees, and we test from both Canada, the FGCA, as well as the HTIRC in the US. Out of 642 trees that were tested, what we found is that there's about 80% of the trees within these orchards that are pure butternuts, and there are about 17% that are different levels of hybrids, either F1s or backcross in either direction. This was also possible through Pauline Hessenauer that did this structure analysis here for us, which was just last week. So happy that she was able to do this so we could put the slide on. So the other part of the project that we're going to be working on is focusing on the landscape genetics of the pathogen itself and trying to look at the quantifying the strain functional differences. This is work that has already been started. We've been scouring the landscape to collect OCJ samples, and this will be done by Bernie Vandermeer. Bernie's a biologist in my lab, and will be doing her postgraduate studies under Richard Hamlin at UBC to look at these functional differences and the genetics of the pathogen. And we'll also be having a lot of collaborators that have already contributed from our collections in the US, such as Anna Conrad, a pathologist at the USDA. So this is my last slide. So in summary, we've been working on trying to build the tools. We're a small lab in New Brunswick, so we have to work with a lot of different people. We have to be friendly people to kind of, because we don't have the expertise in all the areas. But the focus for us is trying to protect the species for the long term. And so from a conservation standpoint, what we've done is we do have an ex situ cryopreservation program that has shown germplasm viability after 7 years. And we're looking at reassessing these trees in New Brunswick because they've already been genotyped. So looking at those trees again and evaluating mortality and tree health will give us an option of how fast that decline is happening on the landscape, which is important to know. We're also collaborating with some folks in the US to do some butternut landscape genomics to see potential local adaptation, because New Brunswick is isolated and disjunct from the rest of the range, and we already have genetic, we know we have 2 groups that New Brunswick populations are genetically unique, so we'd like to know a little bit more to that, because we only have neutral markers such as microsats to support that data set. And from a tree improvement standpoint, well, I mean, we're undergoing OCJ landscape genomics to try to look at virulence and pathogenicity. And we now have a tool to actually characterize our pure and hybrid lines, which we hope to use in concert with some of the orchards that, in the US to try to improve resistance screening and look for phenotypes that could be of interest, such as either chemical profiling or physical attributes of interest. So that's it for me. I'd like to thank our funders as well as all the folks that help support the work that we're collaborating in on the right side. So if you have any time, I'll take any questions.
Thank you, and I can say that Martin, as a dedicated research scientist, I remember when you were doing pipetting with a broken arm. You're trying to do it with the opposite hand. So he's very dedicated to this project. So yes, time for 2 questions. Oh, there we go.
Hello. I was just wondering, is there any work that you do with the public in identifying some of those particular individuals that might not be receptive?
Can you speak closer?
Oh, do you work with the public at all in case you find any individuals that you might want to test that might be resistant to the pathogen?
We're not collecting any resistant stock. We basically are looking at monitoring tree health, but since we're going to have a kind of like a secondary survey looking at what we had from 2014 to 2017 and how that changed over time, it might lead to that. So starting to find trees that are still healthy in areas that are really highly cankered might, pinpoint some interest. The big thing right now is screening for that resistance, and that's why we're looking into strain analysis to say, well, okay, are there strains that are more virulent? Are there strains that are focusing on only attacking hybrids and less butternut? I mean, we don't know a whole lot from that end, but yes, it'd be great to get into selecting some individuals and get into to add to a breeding program. Mm-hmm.
Thank you for your presentation, and I was so surprised that I saw some cryopreservation and then in vitro culture germination, because I am also interested in that in vitro culture, and I have a presentation in the afternoon, by the way. So my question is, even though it has high germination or like some viable assimilation I can see in the picture, but I wonder if there were some comparison with some just seed storage in the deep freezer or like minus 20 degrees germination and like compared to cryopreserved some germination data. Thank you.
So I guess in creating our methods, yes, there's a lot of work that was done on comparing at collection time. So we had some data on that. The issue with endangered species and with nuts in general, we do not collect a whole lot of nuts per sample. And the value of the species and those individual accessions is so high that sorry Dave, we don't want to do 4 times 100 for testing butternut. We don't have it. So we tend to go a little lower. So our values in terms of germinations, we had high germination at collection time point. And we also did a retesting after 2 years and a retesting after 7 years. And viability was maintained. So our percentages were around the 80%, 85% average in using in vitro. And the thing is we didn't stop after the just showing survival of the seed. We actually went past that to full regeneration into trees. So once we're germinated, we have root and shoot. Our success directly into soil and to producing trees is over 95% of what has in our in vitro system. Yeah. No, not quite, not quite. It wouldn't be, I would call it suborthodox. We don't lose viability because they cannot be, because they can be frozen, they can be dried. The problem is that the high lipid content and the oxidation is actually what kills the seed. So we still have to use a cryo. So we're a little at an advantage when compared to oaks, for instance.
Yeah, I was just going to add to that too, is that there was a lot of pre-existing work where we can't store that species in our regular seed bank, so cryo is the best way to extend that longevity and keep it as long as possible. So we're out of time. It's ready for lunch. Brian's going to have a few words to say, but thank you, Martin, very much.
Yeah, thank you. Yeah, just apologies, a little communication error, probably on my part, but so lunch isn't quite ready yet, so you have time to relax and go freshen up, and it'll be next door, and you can show up when the food shows up. Yeah. Well, it was scheduled for now, but the message to the kitchen didn't get through to the kitchen, so they were probably preparing for 12 o'clock. It's a pizza buffet with gluten-free, cheese-free, options for those that requested. But so we'll probably be seeing that show up in the next 10 to 15 minutes. So you can just hang around and then come in and join us for lunch. But we'll try and get back on schedule and start with 12. We'll try 12:40, 12:45 for a restart. So okay.
So a speed buffet.
Yeah, speed buffet. We have a speed dating pizza buffet. Okay. All right. Anyone got a joke? No? Okay. All right. Enjoy your free time.